Good morning. Today we have the seventh of our 6 presentations by the students at the University of Florida. 2 months ago or so, when I was trying to round up students, I had 2 students for 6 speakers. And then I asked for some help from Ken Burns and Jane Brockman and Dan Purich and Jamie Ellis and a couple of other professors. And all of a sudden I had 7 people for 6 talks. So we lucked out, and I think you're really going to enjoy it today. Galen is just back from Tunisia. Maybe we'll find out why Florida bee studies are taking place in Tunisia. But she's going to talk about our bees disappearing in Alachua County. I used to live in Citrus County, sorry. Wild bees may face many threats and have far less help from people than the honeybees. So welcome, and we're glad you're here, and welcome everybody on Zoom.
Happy to be here. So yes, I had the fortune of traveling to Tunisia recently, not for research, for pleasure. But I did see a few bees there, and I was very happy to do so. So today I'm going to be presenting to you all about local bees and native bees. So these are the bees that you might be a little less familiar with. They don't live in hives, but they do live in our natural areas. So who am I? I am a PhD student in the Department of Biology here at the University of Florida, and I've been researching bees and various aspects of their biology for the last 9 years. My focus during my dissertation has been on bee ecology and genetics, and today I'm going to be presenting mostly on the former, on the ecology of our local bees. So if you're here, I imagine you at least have some idea that bees are important and that we need to be thinking about them, concerned about them, so forth. So bees are well known as pollinators. They visit flowers, they spread pollen from flower to flower, allowing those plants to set seed and set fruit. But why are these pollinators so important and so often discussed? Well, bees are incredibly productive pollinators. So they contribute to the pollination of about 70% of our food crops, which I personally am very grateful for. And a large number of crops are actually entirely dependent upon bee pollination alone, including some of my favorites, blueberries, almonds, and watermelon, which I've definitely been enjoying in Tunisia. And one of the things that makes them so effective as pollinators is their unique morphology and behavior. So this clip will be a little blurry, unfortunately, but what it's showing you is a local bee, Habropoda laboriosa, also known as the southeastern blueberry bee, and it's visiting here a blueberry crop. Now, blueberry crops the flowers are pretty unique. So the pollen in a blueberry flower is held very tightly in the stamen of that flower. And most pollinators, when they visit this flower, are not going to be able to access that pollen. But this little blueberry bee right here has a unique behavior that allows it to get that pollen. It'll visit a flower, it'll wrap itself around it and buzz at just the right frequency that allows the pollen to shoot out of the stamen, cover the bee. Now that bee can go to other flowers and spread that pollen around. So without this unique behavior, many crops are actually unable to get pollinated. And one of the favorite agricultural pollinators, the honeybee, does not do this behavior. So it's pretty important that we look at our native bees as important crop pollinators in addition to honeybees. Additionally, bees have really unique morphology. So what you're seeing here is the hairs on a bee. Who knew you would be interested or ever looking at bee hairs? But the hairs on bees are pretty unique. They are branched, and they often have, the branches can vary. They can be longer, they can be shorter. But these branches really help pollen to stick to that bee. And so when they're gathering pollen at these flowers, when they move to the next flower, the pollen is going to be able to stick more effectively, and they're going to lose less pollen during their flight than some of their sleeker pollinator colleagues. So Florida has pretty unique bee fauna. We have about 315 native bee species. The honeybee is not native to Florida. It is the European honeybee and was brought here during the colonial era. But our native bee species are quite diverse. They include all 6 bee families present in the US. So among all those species, they belong to a wide variety of groups. And they also include 27 endemic species. A species is endemic when it exists in one area and nowhere else in the world. And the unique climate of the Florida Peninsula, being the only part of the contiguous US that is considered tropical, allows for it to have these endemic species. The biodiversity trends in the state, however, are inverse to the patterns of endemism. So you find endemic species in the south of the peninsula, and you find the majority of the biodiversity in the north or in the panhandle. Now, bee conservation is a big topic and very popular in the news these days. And both endemism and our biodiversity are important considerations for conservation. You lose an endemic species, there's no other populations to support it. And if you lose biodiversity in an area, you can lose some of the ecosystem functioning that allows these pollinators to help promote biodiversity in plants and other aspects of the ecosystem. Now, threats to bee species are worldwide, and they include 4 big ones: habitat loss, pesticide use, invasive pests and pathogens, and climate change, all of which are impacting our species here in Florida. And when we talk about biodiversity and protecting biodiversity, in a biological context, this includes 2 components: abundance and species richness. So often we think of biodiversity as species richness, the number of species present. But if you think of an environment that has 5 species, and one of those species is present in hundreds of individuals, while the other 4, maybe a handful, that's going to be considered far less biodiverse than an environment where you have 5 species all present in relatively even numbers. And most importantly, a biodiverse ecosystem is considered a healthy ecosystem. And this is because as you have more species, they can provide functions that help promote the existence of all the species within the ecosystem through what we call ecosystem functions. Bees, can anyone guess what their function is? We've talked about it a bit. Pollination, exactly. So as I started my dissertation, I had been studying bees for a few years at that point, and I was well aware of these various threats that are affecting bees. And so I was kind of curious about what the state and condition of our local bees is. Fortunately for me, Alachua County is a good place to be a bee. So bees really thrive in what are known as early successional habitats. So this means that they really enjoy areas that have been recently disturbed, and as a result of that disturbance, they have more sunlight, nutrients are often stirred up and available, and that allows for communities of both perennial and annual flowering plants to arise and provide habitat resources for bees. So common early successional habitats are grasslands and prairies, which if you're familiar with Alachua County, you know we have a few, as well as forests that have been touched by disturbance such as fire. And Florida has fire-dependent ecosystems throughout, including in Alachua County. So fortunately for me, again, there had been a bee survey conducted 14 years ago by a professor at the University of Florida named Dr. Glenn Hall. And so what this allowed me to do was to have a baseline data set by which I could compare with my survey. So I picked 11 of his sites that he had surveyed across Alachua County, and I did sampling in the same time frame of him from February to November. And the sites that I visited were located in Paynes Prairie, San Velasco, and Kanapaha. So roughly in this area, over here, and then to the north. Now, the baseline survey had close to 1,900 bees and found 95 species. And I found a little over 1,500 individuals of 84 species. Now, these numbers, it's important to put into context. What do these mean, right? There's a slight decline, it appears here, but what is the meaning behind that? And when we study biodiversity from an ecological perspective, we break it down into various components. So the first component is alpha diversity, and this refers to the species diversity at an individual site or locality. Whereas the gamma diversity is the aggregate of those sites, and it can be aptly described as the regional diversity. And both of these can be measured strictly in terms of species richness, just the raw numbers, the raw number of species, or they can be measured through indices that account for the relative abundance of those species. Beta diversity, however, refers to the variation in species composition between two communities. So at its heart, it's a comparison of two sets of species. And the nice thing about beta diversity is it can be measured both across time and space. So you can be looking at two communities, one existing here, one existing here, and look at how they differ from each other. Or you can be looking at the same community across 2 points in time. And the thing about beta diversity that makes it particularly valuable is that each of the species in these metrics hold on to an individual identity. When you're doing a species count, each species is a number, 1, 2, 3, but is 1 different from 2? It's the actual identity of that individual species is lost a little bit in those metrics. But in beta diversity, when you're making this comparison, this species holds its identity across the two communities if it's found in both. And so it can tell us a lot about the more specifics of compositional variation that is lost a little bit if you're comparing strictly species richness, alpha or gamma. So I went ahead and started to break down my data a little bit more, learn a little more of what it was telling me. And one of the things that stuck out right away was the abundances. Now, these baseline, they look pretty similar, right? But what I want you to notice down here is the number of days that were spent sampling. So Dr. Hall and I did our surveys a little bit differently from each other, though we maintained the same methodologies. In total, I sampled 320 days over 2 years. He sampled 71 over 4. So it was appropriate to look at the per day differences as opposed to these raw numbers. And what you see is actually a pretty dramatic decline in per day collections. Now, this feels a little concerning on its face. But as with most things in science, you gotta dig a little deeper. So one of the things you might notice is that I have this broken down by bee bowls and netting. Those are the two methodologies used. They're very standard in collecting bees. And if you're trying to get a comprehensive perspective of the species diversity at a site, you want to do both. If you want to know exactly what species are there, you want to use both methodologies because they have individual biases. But if you're trying to do a comparison between two surveys conducted by different people, the value of these methodologies in a comparative sense differ. So, as you might imagine, an individual netting bees might go about it in slightly different ways, even if they use the same net and they spend the same amount of time each sampling event. So there is a strong possibility of sampling bias or observer bias in the netting, despite the fact that it does help us have a more comprehensive understanding of the complete species list. Meanwhile, the bee bowls that were used in this study were the exact same shape, size, the paint, brand, and color exactly the same. The soap brand that was used to fill them with soapy water, exactly the same. So the level of standardization between these two methodologies was different. And thus, I focused on the bee bowl methodology and subsample when making these comparisons. So as we looked at species richness, we noticed a difference from the patterns that were observed with abundance. Here, the species richness, we got a mixed story. Overall, we're seeing a slight increase, but not significant. In the netted, we are seeing more substantial increase. And in the bee bowls, we're seeing a decline. And what I want you to notice here is that the total numbers, the patterns in the species richness are corresponding to the number of individuals caught. Now, one of these matches with the pattern that we saw in the abundance. One of them does not. So the netting appears to match. Let's see, actually, I think, did I put it up here? No, sorry. Appears to match with the number of days. There were substantially more days. There were substantially more bees caught, right? But the bee bowls, the more standardized methodology, the number caught appears to match with the pattern of the abundance declines. And so I'm inclined to believe that this decline in species richness may in fact be reflective of what's happening in the communities as a whole. And when we test this statistically, we find that it's in fact a significant decline. But what you'll notice is, like I said, it matches with the number caught. And this is well known. It's well known that there is a relationship between the number of bees you catch and the number of species you find, right? But up until a point. So this right here is called a species accumulation curve. And it helps us determine how effective our sampling was at determining the true species list of the sites. So what it's saying here is that with the number of individuals you sample, how many species do you catch? And as you catch more and more, up to a point, you're going to have collected them all. And so you add another individual, and you're not likely to get a new species. And so this is where the curves start to level off. So ideally, in sampling, you have all of yours level off. But there is ideal and there is reality, right? And so there is a way to correct for the fact that you're not always going to reach saturation in your sampling, and that's called rarefaction. So what rarefaction does is it takes the lowest sampled site and it essentially forces all the other sites to go down to that level of sampling so that you can have an accurate comparison across sites. And when we rarefy the data, what we find is this decline disappears, right? But what that means is that they're proportional to the abundance declines. So there's not a targeted effect here where one species is significantly declining and others are being maintained. What that means is that the declines that are occurring are happening across, and so you're having a proportional impact to the species richness. And this evidence against targeted causes of declines is supported by the patterns found taxonomically. So among the different families present, you can see that all of them, they declined at different rates, but they all declined. And in a comparison, oh, that's very light. Able to see the gained bar there? Okay. So in a comparison of the species that were gained versus lost, they're relatively even across these different functional traits, because while a cause of decline might be targeted not taxonomically, it could be targeted towards a specific characteristic of a bee, right? But we don't see that. So with all of this, you might be a little concerned. Are our bees declining? And to that I say, just wait. So one of the interesting findings, in addition to all of these different analyses and metrics, is when you look at the overall list of bees that were lost from these sites and bees that were gained at these sites, you'll notice that they are, many of the species on them, not all, but many of the species on them are species that had either been found in other areas of the region in previous surveys, if they were gained, or they had been observed in other databases if they were lost. So perhaps the findings here need to be tempered, because what we're seeing is a shuffling of bees more than a decline. And to explore this a little further, we look at that beta diversity that really allows us to understand the trends in the composition. So this particular finding here, shown graphically, is looking at what's known as spatial heterogeneity. Sounds complex. But what it means, what it's referring to, is the differences between spatially distinct sites. So just the different sites that I was looking at, right? And it was looking at the patterns of these differences over time. So the first thing of note was that all of these sites had relatively high beta diversity, which means that the variation between them is pretty high. The second really interesting finding is that this variation between the sites was maintained over time. So when I looked at these values for the baseline and I looked at them for the resurvey, they were almost identical. So whatever in these environments that is creating variation that's attracting different communities is being maintained, even though those communities are varying over that time period. So different bees are coming and going, but the level of variation between these sites is being maintained. The other thing of interest was the spatial dynamics of temporal heterogeneity. What does that mean? It means that I compared the differences in the same site between the baseline and the resurvey. So over that 14 years, how much compositional change had we observed for a given site? And then I summed that and averaged it across all the sites. And then compared that to a comparison of the entire data sets that are not segregated by site. So this would be the aggregate, the gamma that we talked about, right? The regional. And what I found was that there was substantially more change happening at local levels than at the regional level. And this makes sense, right? So the chance of a small patch of land changing so much that it causes the bee community to change over 14 years is much higher than an entire region of land changing so much that it changes the composition of those bees. Because there is spatial heterogeneity, as we saw in the last finding, right? And so the big takeaways here is declines may be happening. There's some reason to temper our fears over that. And their impact on the larger scale is not quite clear. But what we have seen is that stability in local community biodiversity is likely benefited by the fact that there's variation over space. And that through meta-community dynamics, and what this refers to is simply bees moving from one area to another, allows for the spatial heterogeneity to help preserve community biodiversity. And so you think about it, an individual site might experience a disturbance in any given year, maybe a prescribed burn, maybe a flood, and that's going to change that habitat. But it's also going to, because bees like that early successional habitat, in the long run, preserve the type of habitat that bees prefer. So even though it's disrupting things in the short term, in the long term, it's allowing the type of habitats bees like to be maintained. But of course, that is those events can happen at small scales, which allows for these habitats to be overall preserved across the larger landscape. So that was my first study. My second study, I was interested in one of those threats that we talked about earlier, disease. And one of the reasons that disease is such a concern for native bees right now is because of their interactions with honeybees. So honeybees are known to carry a number of viruses. I think we're now in the high 20s, low 30s for the number of viruses that impact honeybees alone. And these viruses have been spreading worldwide. So a lot of them have been identified in specific regions, but we're now finding them pop up as honeybees have spread worldwide. And so what you might recognize or not recognize here is a virus that we are probably all very familiar with here, COVID-19. And COVID-19 shares a similarity with honeybee viruses. And that similarity is that, like COVID-19, honeybee viruses are able to cross the species line, right? We heard about COVID happening in cats, and I believe it was ferrets. And so we know viruses can transmit cross species. And this is a big concern because these viruses can have some pretty severe consequences. The most well-known of these viruses, deformed wing virus, creates physical deformities. Black queen cell virus and sac brood virus decimate brood, which are the larva and the pupa of bees. An Israeli acute paralysis virus causes behavioral abnormalities, such as shaking, that is so severe they cannot fly. And in less severe versions, they lose their ability to navigate. So the interactions because honeybees are becoming more and more prevalent in the broader environment as we use them for agriculture, as we have them in our backyards, their interactions with native bees increase and allow for potential host shifts. So the transfer of viruses cycling within one species to start cycling in another. And the basic principle behind this is you have a lovely, cute little honeybee landing on a flower, but it's depositing virus. And then you have a native bee, maybe a bumblebee, come and visiting that same flower and picking up those virions, moving along its way. And so a lot of research has started to develop to try and understand this process of cross-species transmission, how many of the native bees it affects, what native bees it affects, if it affects them. And then, have we achieved true host shifts where this virus is able to replicate and actually infect native bees? Or is it simply a matter of spillover in which the virus that's present in honeybees occasionally gets eaten or gets consumed or otherwise becomes present in native bees, but simply as a matter of spillover, not as a matter of endemic or internal transmission among a different host species? There's a lot of questions here, and we don't have all the answers yet. But I tried to provide a few. So my study, I went out to those 5 of the same sites that I sampled before, and I collected 350 bees, and I tested them for these viruses. And I was looking at the differences in native bees in blue and honeybees in yellow. And we see that there's definitely some dominant viruses here, BQCV and DWV, both of which were in very high prevalence in the honeybees. But they're also in higher prevalence in the native bees. I was also able to measure titer. So this is the level of virus that's present in the bees. And you notice that it's generally higher in the honeybees. In fact, the median titers are always higher. But there's quite a lot of variation in the levels of titers among individuals who had detected positive. And so part of the question that I will hope to answer moving forward is, what do these higher titers mean? Are they infections, or are they just they consumed more? Another really interesting aspect of this sampling is that I was able to look at viral titers over a season. So they're collected within 1 year, but across seasons. And we see that there are spikes, at least in the honeybees, for DWV and to a lesser extent BQCV in the fall. The same pattern is not necessarily observed in the honeybees. Another really interesting aspect that gets at this question of whether we're seeing a host shift versus spillover transmission is that these native bees detect positive for the viruses in the absence of honeybees. So these green bars are measuring on a given sampling day the level of the relative number of foraging honeybees present. And you'll notice in May, there's an absence of honeybee activity. This is normal. The honeybees tend to be very active early as they're building their nests and trying to build up stores. And they're also really active in the fall as they are trying to build up prior to the winter. So there is some seasonal variability. Keep in mind that these sites are in natural areas, so there's not an expectation of honeybee colonies that are managed being nearby, right? So the honeybees I'm observing are likely feral. But yeah, their activity is absent, and yet these native bees are still detecting positive. Additionally, because of the way I collected my presence data for these viruses, I was able to look at co-occurrence because each individual bee I tested was tested for all 4. And what we see is that in addition to having really high prevalence compared to the other groups, Apis mellifera are the honeybees, by the way, and these other groups are native bee families. We also see that there is differences in the relative grouping of these viruses. So we have far lower co-infection or co-occurrence in the native bees than in the honeybees. And you'll also notice there's a much greater proportion of individuals with no detection. Now that's pretty curious because there's 2 different ways that might come about. All of these bees were found foraging. They're active. They're not so sick that they're lying on the ground, right? And if a bee who is well enough to be foraging is showing detection for virus, that means that bee has a pretty good tolerance. So disease tolerance is the ability to manage disease symptoms without affecting the actual ability of that pathogen to be present or to replicate or to do its thing, right? That's disease tolerance. But there could also be barriers to host shifts. From the perspective of the virus, this bee that it's entering is a new environment, right? It's used to the honeybee. It's been in the honeybee bunches of times. But when it's entering into a new species, it's a new environment for that virus. And like any novel environment, it could be friendly or it could be hostile, right? And so there could be potentially barriers that are preventing effective host shifts and allowing, and thus we're observing greater proportions with no detection. So it's not clear which one of these is happening. More work certainly needs to be done. This is definitely a preliminary study. But it is interesting to see that there are differences, and it's very interesting to see that there is presence of virus in the absence of honeybee activity. My next direction is to look at the difference between detection and infection. So you'll notice I said things like co-occurrence and had to correct myself about co-infection. Because in detecting these viruses, the method by which I detected these viruses shows that they were there, but doesn't necessarily indicate that they were inside the cells of the bee causing infection, replication within the bee. So you can potentially consume a virus and it could pass through your system completely harmless. But to cause an actual infection, it has to be able to enter into your cells and replicate within them, and potentially cause disease, right? Well, all of these viruses are what are known as single-strand viruses. So within their mature state, the genome present inside them is a single-stranded RNA. Well, in order to replicate within a cell, it has to go through something called double-strand RNA synthesis. It has to go from being that single-strand state to a double-strand state in order to produce the proteins that will create new virions. So to infer infection, one way you can do that is you can test for the presence of the other strand. And that will be my next step so that I can go from just talking about presence within these native bees to hopefully informing the larger research community as to whether these bees can be truly infected or not. But that is for another day. Thank you so much. I apologize. No, I didn't go long. Yes. And I would love to open up to any questions or thoughts.
Thank you very much. Do we have any questions? I see one in the back.
Yeah, so I noticed that you took great care to create the same kind of collecting conditions as the previous endeavor. And I'm wondering if there is any changes in the bees that could be affecting the changes in the final data. For example, could they be learning from the experience of getting caught in these cups or swept away in a net? Could that learning be spread among each other and passed through generations? Or such as you notice in the north, deer at the time, as soon as hunting season opens, man, those deer are in the trees. They're not out and around. And they've obviously learned something about being collected, so to speak. So anyway, I was just wondering if you had any thoughts on that.
Thank you so much for your question. That's a really interesting idea. And I'm going to touch upon it in a few different ways because I don't believe I have the full answer to it. So bees are known as social creatures, right? Especially the honeybees. We know that they are very intelligent and they do learn from one another. The learning that has been shown in honeybees has only been shown within generation, or it can be across generation, but it would be like a sister bee reaches maturity and then learns from her older sister type deal. So in honeybees, a reproductive event for a honeybee is thought of a little differently than a reproductive event for us. We think of having a child as a reproductive event, right? But honeybees are considered superorganisms because of their social structure, and so the reproductive event for a honeybee is a colony splitting. And so as the colony splits, potentially there's room for learning across that split. But I think the level of learning over generations is probably limited. The other thing that I'm going to mention is that the majority, over 80% of bee species are not social. I like to refer to native bees as the hardworking single mothers, because a lot of times the females will build their nests, lay their eggs, and then they're done. So there's actually no interaction between parent and offspring in that case. And the learning within, and they're also not interacting with each other within cohort the same way that the social bees are. So over the course of 14 years, it's unlikely that they would have learned enough to change behaviors and avoid these collection methods. Additionally, these methodologies have been shown time over time in the larger entomological world as being successful, even when done at same sites. But I think that is an intriguing possibility potentially within season and for some of those social species. Yeah.
I have a couple of questions. I'll wait for you. Okay, one of my questions is the variation in the vegetation in the different sites. Now, did you keep a map of the aggregate stuff that was growing 14 years ago and how it changed and how that may affect your numbers?
Absolutely. Good question. So neither Dr. Hall nor I took a comprehensive plant survey during our surveys, but what we did do was we took note of every single plant species that we netted off of. So if we collected a bee, that bee got identified by its taxonomic identification, its species and genus, as well as, family and so forth. Its sex, male, female, what plant it was taken from if it was netted, or what color bee bowl it was found in if it was found in a bee bowl. So we do have some information regarding the plant communities. And one of the reasons I didn't include it here, although I think it's very interesting and there's potentially room for a second publication that I would do this type of work in, I didn't include it here because it's not comprehensive. So just because the bees that were netted off, or the plants that were netted off of, are invariably going to be a subset and maybe not even a complete representation of the plants the bees are visiting for resources. And so I felt, given that limitation of the data, it may not be possible for me to do that type of comprehensive analysis. But one thing I could do that might be interesting for another paper is to look at changes in the visitation network, is what we call it. So when you're collecting this netting data, you're collecting the patterns of visitation across the community. And looking at the change in that visitation patterns over time could be really interesting. But I do believe the difference in the netting sampling may be a limitation there for how much conclusion I can draw about the change over time, simply because he did so much less sampling. So there is a chance that he missed more of that netted community.
Thank you. And then another question is, these native bees are not colonies, so they're not collecting honey. They're just drinking the nectar for their own health, and they don't take it back and give it to anybody else. They don't share the pollen. Their collection of pollen is not for the honeybee, whatever it is they give to the queen. So it's just an accident of where they are and what they're doing.
So that's a good point, and it's not quite an accident. So native bees do intentionally collect pollen to feed to their young. And native bees vary in terms of whether they're specialists or generalists. So some native bees will be collecting a wide variety of pollen and providing it to their young in a big pellet. Or there are some bees that are specialists that require very certain flowers to visit and collect that pollen, because that pollen is ideal for the development of their young. So there's great variety in terms of the importance of plant diversity for a given species. But yeah, no, they're very intentional about pollen collection. You are absolutely correct about the nectar. They are not creating honey. They are simply, when they're going to visit a plant for nectar, they're just trying to get some good sugar nutrients for themselves.
First, I'd like to compliment you on, you answered a lot of my questions. Admittedly, I'm a mammologist, so my collection's a little bit different.
Fair.
But one of my questions was about the missing species and what they have in common, and you did that.
Yeah.
You went, that was very good. And the diversity of the sites. I have a couple of questions. I also would like to see more on the plant species that were there, and I had to become a botanist to be able to do that for mine and identify them. And I think it's really important.
Yeah.
Also, I wanted to make sure that you were netting the same way.
Yeah.
And how did you, I want to know how you chose the 11 that you chose to do.
Yes.
And then also, how did he originally choose them? I know for me, some of mine was that I had to pick areas that were either part of a park or privately owned, the kind of thing that wasn't going to disappear next week.
Yeah.
So, I mean, you may have been only able to go back to the 11 because they were the ones that were available.
Yes.
And, but I'd really like, there was one more question, which is now gone from my brain. But I'd like to see definitely the species, because if they've changed, then that may be why some of yours have moved
Absolutely.
away.
Absolutely. No, that's a very good point. And I think that is one of the challenges with the data set, and definitely helps to explain why it's been a little hard to explain the declines. And why I think it's also fair to be a little cautious about how, like, that 88% seems very extreme, but I think it's worth mentioning that some of that variation, some of that potential, what looks like a decline, can be explained by other sources of variation and is not necessarily all attributable to a decline in population. But yeah, so the plants, I agree with you there. I think that was a limitation. The reason, so he did originally 16 sites and I did 11 of those. I started sampling in February 2020. The pandemic hit. It changed a lot about my work, and as a single individual, I also sampled more heavily than he did. And so for a single individual, I simply could not go to all the sites at the frequency I had hoped to for. So his sampling was done with a very different intention than mine. Mine was very much done to provide not only a comparison to his baseline, but to provide a future baseline. And as such, I sampled in a very systematic way that was not necessarily reflected in his sampling, but the methodologies by which we sampled were the same. So, yeah, and so I simply couldn't go to all of them. And some of them were on private property that I didn't have access to, although I was very fortunate to get access to a private preserve. Some of them, it was feasibility. So I was hauling these big stands for putting my bee bowls on, and I, as a single individual, just did not have the strength to carry them to some of the sites. So I had to find the sites that were more accessible. So yeah, science is always going to be limited, unfortunately, sometimes by logistical concerns. But the data set that I compared to was accounting for that. So I didn't use his full list. I subset it by the things he found at the sites I resampled. So the comparison was a direct one-to-one comparison.
Any other questions? Is there anything on Zoom?
No, but I do. I have a question,
Yes.
if I may. Do you have any information on the rise of area temperature or water temperature in these places that could affect or influence the prevalence of the viruses, the increase?
Ooh, yes. So I did record environmental conditions every time I went out. I thought you were going to ask about the biodiversity, because that's where I was focusing it. I have not thought about doing environmental analyses, in part because the viruses were collected only now. So I can't look at change over time with the viruses. There's only a single time point collection for that. Potentially look at it across season and how seasonal differences might impact that. But as far as environment and the biodiversity, I'm still in the speculation stage, so I hate to put anything out as definitive conclusions. But I sampled in very wet years and also years that experienced some of those disturbances. I had controlled burns go through my sites. I had floods at my sites. I had wild animals grazing at my sites. So those things could certainly impact whether bees want to be present at those sites in a given year, right? But I think it's important to recognize that those activities are longstanding. Those same activities were occurring when Dr. Hall was sampling as well.
Okay, well, thank you very much. Let's give her a big hand for the 7th out of 6 speakers.
Thank you all for having me. I really appreciate it.
I think we had an outstanding 7 speeches or talks over the 7 weeks, and I appreciate you answering me from Tunisia saying you couldn't make it the next day. So thank you very much.